So thank you for the introduction. I'm glad to have been part of this group more recently and have been learning a lot from these seminars. So I'm happy to tell you a bit about what I've been working on in my lab. So I'm going to start out with background on Gardnerella and the urogenital niche. I'm sure many of you are familiar with it because it pops up in a lot of Urobiome data sets, but Gardnerella historically has been recognized as a common member of the vaginal microbiome, where it has been linked with a condition called bacterial vaginosis, which is one of the leading, most common vaginal conditions in women. Roughly 1/3 of women at any given time have BV. And while it can be asymptomatic, it also, regardless of whether it's symptomatic or not, it's associated with increased risk of other urogenital infections, including urinary tract infections, which is what kind of got me into the urinary tract field, actually, because I started out studying Gardnerella in the vaginal niche. Oh, I guess I have to use the arrow to move my slides. All right. So in the urinary tract, Gardnerella is a relatively rare cause of actual UTI, but as many of you are familiar, it's a frequent member of the urinary microbiome. And some of the initial papers that came out with the urinary microbiome reported that in some instances, it was more frequently isolated in patients with a history of either recurrent urinary tract infections or urgency urinary incontinence. And that has held true in other studies since then, but it hasn't always been the case. And a lot of what we are working on is try to understand why that may be. So we'll get into that later. But I want to walk you through a little bit of the history of Gardnerella. So the role of Gardnerella in pathogenesis in the urogenital niche has actually been the source of debate for ever since it was first identified. So back in 1955, Gardner and Dukes isolated Gardnerella. Back then they named it Haemophilus vaginalis, and they said, this is it. This is the cause of what back then was called non-specific vaginitis. Now it's the same thing that we call BV. But moving forward through the years, people were, appreciating that Gardnerella is not the only organism present in BV. While sometimes it can be the most abundant, there are a lot of other anaerobes present in this polymicrobial condition. And so the question was being raised, well, is Gardnerella really the pathogen in BV? 30 years after isolating it, Gardner still doubled down and said these other co-pathogens, they don't matter, and Gardnerella is never commensal. It's the pathogen. But this doesn't really fit right with data using more molecular techniques, where even women who are BV negative, more than 80% of them have detectable Gardnerella by PCR. So the debate continued over the next years. A lot of questions about whether Gardnerella is actually pathogenic during bacterial vaginosis, and if so, which strains. And so 60 years, and actually I looked back, I actually stole this slide that I made during my postdoc. We're now to 70 years since its first isolation. McDonald's has substantially improved its cheeseburger, but we still are asking, is Gardnerella vaginalis really a pathogen? And one of the reasons that we're still asking that can be linked back to even what we call it. So again, this is a slide I made during my postdoc. Gardnerella vaginalis seems pretty simple. That's what all Gardnerella were called. There was only one species. But Gardnerella actually has a very complicated taxonomy, and the genetic diversity between strains is really high. And we've known that for a long time. And over the years, different groups have come up with different ways of parsing out Gardnerella based either on in vitro phenotypes or getting into more molecular techniques. You see that Gardnerella is being divided into all these different ways. And as we get into full whole genome sequencing analyses, it was first divided into 4 clades, then ecotypes. Then in 2019, some new species names have been proposed and are at play now. And as you can see, that still hasn't closed the debate, and we still have other groups coming up with other nomenclatures and other ways of slicing and dicing the Gardnerella pie. But what we're still really left with is, are they all, and which ones are pathogenic? And we know from the clinical literature that BV is not always symptomatic, so you can have some of these Gardnerella strains there and not have symptoms. And we see inconsistencies both in the vaginal microbiome field, as far as looking for associations with preterm birth or sexually transmitted infections or with LUTs, UUI, that not every study finds Gardnerella associated. And one of the reasons that we think that could be is because 16S sequencing can only distinguish Gardnerella at the genus level and fails to capture this genetic diversity between strains that probably play a key role or that probably contribute to different pathogenic outcomes. So I've been studying Gardnerella for a while now in the urinary tract, and I focused first on the potential for Gardnerella to affect urinary tract infection by uropathogenic E. coli. And so I'm not going to go through the data from any of this, but basically we found that if you, in mouse models, give mice an inoculation of Gardnerella, either before you give E. coli or after you've given E. coli, it makes an E. coli UTI worse. We see increased acute bacteriuria. We see increased levels of persistent infection, higher bladder titers in bacterial reservoirs, and also Gardnerella can trigger E. coli to come out of reservoirs to cause a recurrent infection. Now, all of those studies I did with the one strain of Gardnerella that was a vaginal isolate from a one with BV, and it's a clade 2 strain, or now its species name is GPODI. I'm going to only refer to my Gardnerella strains at the clade level because too many different names doesn't really help our cause in this situation. So the hypothesis that we've been interrogating for the last year and a half or so is relatively straightforward. It's that the pathogenesis of Gardnerella in the urogenital niche is going to be clade or strain specific. And Lokesh Kumar is the staff scientist in my lab, and he's been doing this work. And what he's done is taken our mouse model of urinary tract inoculation with Gardnerella, and he's comparing strains from across all 4 of the major Gardnerella clades. We set up this timeline to match what I had done in our urinary tract infection model, whereby we need to give 2 inoculations with Gardnerella. The reason we needed to do that is because Gardnerella actually got cleared from the mice relatively rapidly. So the strain that I tested before, by 12 hours, most of the mice no longer have bacteriuria. But even though the bacteriuria didn't persist, we still saw evidence of urothelial disruption, and it still impacted the E. coli UTI. We've been calling this covert pathogenesis because it's a way that Gardnerella can promote pathogenesis by another organism, but it's gone and you wouldn't even know it was there by the time you see the phenotypes. So Lokesh has been inoculated mice 2 times with these different Gardnerella strains. There's no E. coli in this context. We're just interested in what is the effect of Gardnerella on the bladder tissue. And then he collected urine and tissue at distinct time points, and then we looked at what we saw. This is a phylogenetic tree of the strains that he used. So we tested 18 Gardnerella isolates. These are all clinical isolates. They came either from vaginal swabs or from urine samples, a couple of endometrial isolates. And you can see that we have strains from across the 4 main clades. We have fewer of clade 3, but that's consistent with what's in the literature. There's fewer clade 3 have been isolated and sequenced. So I'll show you the bacteriuria data. So on the left is 18 hours after a single exposure. On the right is 6 hours after that second exposure. And what I think you can see right away is that it's a couple of clade 2 strains were able to persist better in the urinary tract. Each shape is a different strain. So we had 2 strains that behaved differently than what I had seen before. I was actually not really expecting to see any of them persist this long, but these 2 were able to persist in the urinary tract for 18 hours. But most of the rest of the strains were non-detectable. We thought that maybe urine isolates would do better than vaginal isolates, but that's not what we saw. We only one of these strains that had bacteriuria was a urine isolate. The rest were vaginal. And so I mentioned that we had shown that Gardnerella causes exfoliation. This is what that looks like on a scanning electron micrograph, where these superficial urothelial cells have exfoliated away, and now you can see these underlying transitional cells. A better and more quantitative way that we could look at this when comparing our strains is to stain sections for cytokeratin 20, which marks the terminal differentiated umbrella cells. You see this nice bright staining along the lumen. And in mice that are exposed to Gardnerella, you see patches where that staining is gone because those cells have exfoliated away. So Lokesh did staining of bladder sections from mice that were exposed to all of our different 18 strains. So just to orient you to this data set, Here, cytokeratin 20 is stained in red, and so you see this nice red line lining the lumen. Chitosan, we know, triggers exfoliation of virtually the entire urothelium, and so you don't see that nice bright red line. And we saw a varying degree of exfoliation between our different Gardnerella strains. So some of them, no exfoliation was evident. Some of them you see more, patchy exfoliation. Some of them we see looked a lot like our chitosan positive controls. So we had two researchers blindedly score this exfoliation from these sections. And what we could see at the strain level, there were some strains that caused significant increase in exfoliation. And when you combine these data all together by clade, we see that the clade 2 and the clade 4 strains were the ones that were able to cause epithelial exfoliation in the bladder. Another observation that we made in these bladder tissues that was kind of unexpected is we saw that some of the strains caused pretty substantial edema in the tissue. So hopefully what you can appreciate right away, like right here in this 8151B strain, this clade 2, these clade 4s, all that white space in the lamina propria is edema in the tissue. And it's not something that even a chitosan exfoliated treated bladder doesn't look like this. Similar to the exfoliation results, it was the clade 2 and clade 4 strains, both that caused edema, and also one of the clade 3, but not the other. We saw this phenotype. And just as a little bit of a side note, because I don't have a lot of follow-up about this phenotype and what else is involved in this response, but it looks a lot like a bladder section from mice in models of interstitial cystitis. So this one here I pulled from a paper that used cyclophosphamide-induced IC. And if you look back in the literature, there are a few papers that have looked for Gardnerella in IC patients. One found it detectable in biopsies. Another more recent one found Gardnerella more often in patients with Hunter lesion IC compared to non-Hunter lesion IC. But yet again, another paper looked for Gardnerella and didn't find it in biopsies. So this could be another instance of covert pathogenesis, and also these previous studies only looked at the genus level. So the discrepancies between these data could be at the level of like sort of which strains are involved in this phenotype. So at the end of all these mouse experiments, we have this sort of summary of phenotypes. So to summarize these, we've determined that the ability of Gardnerella strains to cause pathogenic effects in the bladder isn't really related to whether they came from a vaginal swab or urine. Persistence wasn't required to induce exfoliation or edema, so plenty of strains that we never detected by bacteriuria, we still saw that they caused that damage. We thought that maybe strains that cause more exfoliation, actually maybe that helped flush the bacteria out of the bladder, and we might see that sort of an anti-correlation there, but that's not as clear. And then also exfoliation and edema appear to be independent. So some strains caused one and not the other. So if you were studying a model organism, sort of your next step with this kind of data would be to do some sort of, reverse genetics and try to identify which genes are responsible for these phenotypes. But many of you are aware of what it's like working in a fastidious urogenital organisms, and we don't have those tools available. Gardnerella is not genetically tractable, so we don't have a way to do that. But I'm often telling my kids that having friends is more important than having tools or toys. So it's good when you have a friend that works just down the hall who is an expert on comparative genomics, and he also happens to love Gardnerella, which is great. So collaborating with Dr. Robert Potter, who now is at CHOP, we were able to take the whole genomes of these 18 isolates, and we parsed them into whether or not they elicited these particular phenotypes. And then Robert puts them through a pipeline where we can look for which genes were associated with these phenotypes. And we can do this because the accessory genomes are quite distinct between different strains of Gardnerella. And so after Robert did his genome comparison magic, we ended up with 45 genes that were significantly associated with one of these phenotypes. And the way that we are trying to represent this right now, you're not meant to sort of read all the nitty-gritty over here, but I'll just kind of walk you through what we saw. So 7 genes were associated with colonization, which is what we called that being able to persist out to the 18-hour time point. 34 genes were associated with the exposure, so they were still present at that shorter term bacteriuria, and then a couple of genes associated with the edema and the exfoliation. And some things to just point out is 3 of these have LPXTG motifs, suggesting that they're surface exposed and could be interacting with the host. 5 of them are carbohydrate metabolism. We have an extracellular matrix binding protein. Most of the COG categories, the most prevalent COG category was cell wall membrane and envelope biogenesis. And, now that we've got this gene set, this opens up a lot of directions that I don't have data for yet, but we're excited to have these results. And we're thinking about these genes more as potential biomarkers for uropathogenicity of Gardnerella strains. And so we wanted to look at how conserved these genes are across all Gardnerella, and can we see patterns in that data. So we took this list. Robert, I see it was Robert. Robert takes this list and he looked at the pangenome analysis of 291 publicly available Gardnerella genomes and looked for the presence or absence of these genes. And so what you get is this big, beautiful wheel, and I will walk you through this because I know there's a lot to see here. So in the middle is a phylogenetic tree, and any strain that we tested in this study has its name, poking out the side there. Each spoke on the wheel is a strain. You see that they've divided into the 4 main clades, and then each square is, if the square is there, that gene is present in that strain. And we've color-coded the squares based on the phenotype. So in purple in the middle are those for persistence, and then edema, exfoliation, and exposure. And so some things that you can notice just by gazing at this, I hope you can appreciate that persistence and exposure genes, so the purple and the gray, are conserved more in the clade 2. See a lot of clustering, especially of the purple in the clade 2 strains, suggesting that, and both of the strains that we saw that persist were clade 2, so suggesting that clade 2 strains might be more capable of persisting in the bladder when they make it there. Both exfoliation genes were conserved across nearly all clade 4 strains. There were only 8 strains that didn't have those exfoliation associated genes. And the clade 3 strains are largely lacking most of these genes. So to summarize some implications of the results that we have so far. So we're able to say now that most Gardnerella really don't persist in the urinary tract for very long, and that there are strain and clade level differences in the pathogenic phenotypes elicited by Gardnerella. Clade 2 and clade 4 appear to be more pathogenic, at least to the urothelium and the bladder tissue. Clade 1 strains, we didn't ever see a damaging phenotype with any of our clade 1s. I think that this has implications for how we move forward, potentially screening for pathogenic Gardnerella, reminding that, when and where we look and which strains we look for are going to matter. And another thing to close with, and that again opens the door for future studies, is all of these phenotypes and characterization that we've done were done in naive young mice. So they have a healthy bladder essentially. And we know that there are a lot of situations that the bladder could be more vulnerable. And it's quite possible and likely that some of these Gardnerella strains that weren't pathogenic in a healthy naive bladder pathogenesis could be revealed in the context of like an aged bladder or the pregnant situation, or if there's a history of prior infection, so the bladder has been remodeled. So we're excited to sort of explore that further in the future. And with that, I want to stop and just acknowledge everybody in my lab. Lokesh Kumar did this work along with some help from Sonia and Debbie. I want to thank my funding sources, Robert Potter for all his genome magic. I got strains from my previous PI, Amanda Lewis, and Alan Wolf, who I'm sure you all know, and Garth Ehrlich. And yeah, I would just be happy to take any questions. Thank you.
All right, well, thank you. Well, I think that this is a great group of people to get some feedback on. So I'm looking for feedback on a number of things. My talk today is going to be on a slightly different topic than what we've usually convened around, which is stories about microbial ecology and how we interact with our urobiome. So today I want to shed light on a slightly different facet of the ecosystem, which is the biochemical aspect of the ecosystem and of that, particularly in postmenopausal women. So we've been studying the postmenopausal urobiome in the context of recurrent UTI now for a while. And I don't need to belabor this for this audience, but recurrent UTI and UTI are certainly a global health problem. And we really are trying to understand how this cycle of infection can be intervened and perhaps one day even cured. In postmenopausal women, it's a very large problem. The epidemiological data is not always super tight, but some reports saying up to 50% of UTIs in postmenopausal women become recurrent. Most of our treatments rely on the use of antibiotics, first-line antibiotics. We're starting to see a lot of resistance in clinical isolates and in metagenomes that we sequence. And ultimately, these lead us to conclude that these are the current treatment options are unsustainable, and they don't take into account the fact that we know that there is a resident microbial community present. A resident microbial community that might, and we believe now with evidence is coming out from many groups, is related to health and disease. And one of the major opportunities in the field right now is that there's really no options for personalized approach to management. One UTI is not the same as another UTI, especially in the case of recurrence, where the history of this disease and this infection and its treatment begin to shape the host in ways that we have not really measured yet. Urinary tract has a resident microbiome. We don't need to really go over that too much here. It's why we're all convened. You know, we work a lot with this is just chromogenic agar plating 100 microliters of urine, and we can get a bunch of pretty colonies on it. But we're beginning now to recognize that the urobiome is something that we need to consider in the management, particularly of recurrent UTI. And we've spent a lot of time I got into this field about 6 or 7 years ago now. And since that time, I've spent a lot of time asking questions about the urobiome, particularly its ecology. So what is the ecology of the urobiome? How is it modulated? And can we influence the urobiome to protect from recurrent urinary tract infection? And all of this, to my work so far to date, has been predominantly in the postmenopausal space, as they are the most affected demographic by this disease. So in about 2018, we set out to study this in a human cohort. We worked with Dr. Philippe Zimmern at UT Southwestern, and we designed a cohort that mimics the UTI pathogenesis as best we could in a cross-sectional format. So we have these periods of infection followed by intervening periods of where is presumably not infection, but let's say perhaps their symptoms subside. But in our cohort, the in-between infection cohort, a group of women are culture negative, and another infection. So our cohort really has 3 groups in it, with 2 of the groups having a recent history of recurrent UTI. They're actively seeking high-level management of their recurrent UTI. They differ by the fact that one of them is UTI negative at the time of urine donation and one of them is positive. And then we also have a healthy comparator group of postmenopausal women that are no lifetime history of UTI, no evidence of lifetime history of UTI. And so from this group, we've published a couple of papers on it, and we've done an extensive amount of work characterizing this cohort. Not just myself, but also many other people in the DNISCO lab, where we have our 3 groups. And from this cohort, we did whole genome metagenomic sequencing on the urine to capture the microbial profile, but also the functional profile, the genetic enrichment that is present in that microbiome. This is a highly phenotyped cohort, so we have a lot of clinical metadata. And we also have a bacterial and fungal isolate biobank that we go back to frequently to test hypotheses that we make in these large associative studies. And so basically what we observed so far has been stories in microbial ecology. The 2 major stories that we've published on are that in this cohort, when we look at our 3 groups, the groups that have this history of recurrent UTI have an increase of what we would associate with dysbiosis. Some taxa on a profile associated with dysbiosis. Taxa are present there that perhaps should not be there. And they have increased antimicrobial resistance genes, even in the absence of infection. This group that's in the middle of kind of their infectious cycle, they have an increase of antimicrobial resistance genes in their microbiome, even in between infections, in the absence of infection. These women that have no UTI history have increase of what we believe are protective lactobacillus and seemingly the lack of antimicrobial resistance genes in their urobiome. So this story really has, when I think about the things that we observed here, it's really a story of microbial ecology. Who is there and what do they have? We also noticed a strong association with estrogen that I'm not going to talk about today, but we found a decreased diversity and strongly increased abundance of lactobacillus when these women were taking estrogen hormone therapy. But microbial ecology honestly can only get us so far if we just focus on the microbes that are present. If we break everything down to a list of the features that are in the microbiome, we have E. coli, lactobacillus, we have all of these other taxa that are present. If we boil all that context of this perhaps very vibrant living ecosystem down to just a list, we miss a lot of the context. So I want everyone to look at this. This picture is a picture of an ecosystem where there's several species present. And if I were only to annotate that there were zebra present, that there were giraffe present, and there were elephants present, and I tell you how many they were, and I told you the proportion of the population was represented by zebra, giraffe, or elephants, we would miss a lot of context about the ecosystem. We would miss the fact that there seems to be a nutrient source for these zebra here in the foreground, that these elephants like to be back here and maybe eating some grass or something. And it's not really a whole lot to say about this giraffe, except that they don't really like their picture taken. But we would miss a lot about this ecosystem if I only enumerated a number of species present. So when we're doing if we take that analogy and we look back at the microbial space, we usually are working in the space of DNA and counting basically the number of species present using metagenomics. But if we begin overlaying other types of omics-based approaches, we begin getting closer to biological context. Certainly not a full picture, but a little bit more context. So what we do is we've really spent time in the small metabolites that are present. We call that the biochemical space using metabolomics, and then looking at the biochemical ecology. So our current work is integrating both microbial ecology and biochemical ecology of the postmenopausal urobiome to understand a little bit more context about this ecosystem. What I like to say is these are the things that our favorite sequencing technologies can't tell us. We can get a list of species present perhaps, but we can't get their context. We can't understand, for example, what nutrient sources they might be correlating with. Still only associative analyses, but we can get a little bit more context by integrating other types of enabling technologies. And in this case, we use mass spectrometry-based metabolomics. So what we've done is we've gone back to this same matched cohort that we've published on before, and we've performed a large metabolomic screen using a commercially available kit from a company called Biochemical that measures about 630 different metabolites. There's a little bit of chemistry that goes on, and you have to have a mass spec available, but you get absolute abundances of 630 different metabolites. And here's just a representation of the coverage of the type of metabolites that we got, all the way from, these are just in alphabetical order, acylcarnitines, amino acids, biogenic amines, all the way down to things like triglycerides, which we did not really observe very many of because urine is an aqueous substrate, and these are very nonpolar molecules. But we got relatively good coverage. This is just the percent of the cohort that was above the limit of detection for an individual metabolite. So from this data, today I want to discuss basically two things that we asked from this data. We asked a fundamental question about how the urobiome, the microbial ecology, associates with the local biochemistry in urine. And then we also began asking very translatable questions. Is that now that we have this joint information about the urinary biochemistry and the urinary microbial communities, can we use this information to find any biomarkers or anything that might help us move towards a more personalized approach for recurrent UTI? So we'll talk about this first one first. And I really want to kind of drive home this idea of we're studying an ecosystem using ecological means. There's a whole set of mathematical approaches to study ecologies. We can look at things like beta diversity, alpha diversity, gamma diversity. I've never really used, but alpha diversity being the diversity of the sample, how diverse a particular sample is. And beta diversity being how different are different samples from one another. So we can ask a very simple question. Are samples that are very taxonomically different from one another, are they also very biochemically different from one another? So we have information about the biochemical beta diversity, basically distancing samples from one another based on how differently they are biochemically in their metabolome, and how microbially different they are from one another. And we can just do some statistics to understand. And we looked at all of these basically distant measures between the metabolites and the taxa. And what we found is a significant but relatively weak, actually, the absolute association between the microbial diversity and the biochemical diversity just in urine. What we did notice was as we started going down the list of abundant metabolites, we started finding that high abundant taxa seem to be associated with low abundant metabolites. And that starts to make a little bit of sense in this case, because this is a relatively low biomass microbiome, and most of the metabolites in urine are probably going to be related to the host, whether that be through urinary excretion or diet or local inflammation. Another interesting signature we found is that we found two metabolites, which were methionine sulfoxide and trimethylamine oxide, that were relatively strongly correlated with microbial alpha diversity, meaning the abundance of the local microbiome. So as the microbiome became more diverse, meaning as more species were present in the microbiome, we saw that these metabolites, which are associated with local oxidative stress, also became more abundant in the urine. So one of the things I'd like to follow up on eventually is testing that hypothesis that are we seeing signatures of dysbiosis in the bladder or local inflammation that might be associated with the microbial ecology, and hence we're observing a signal in the local biochemical ecology as well. And then we can also look at things like the overall topology of the taxa metabolite network. So I'm going to show a network where we have nodes that are either taxa or metabolites, and the edge, the connection between them just being the correlation coefficient, essentially, between those two, just taxa and metabolites. There's no taxa-taxa nodes, there's no metabolite-metabolite nodes, just taxa metabolites. We can look at the positive association network that we see in our data, and there's not really a whole lot of very gleaming information that you can get from this, except that there's one anomaly down here at the bottom, one taxa that seems to be associated with many, many metabolites when it's present, and that is uropathogenic E. coli. In this data set, UPEC was the most prevalent pathogen in the data set, as it is in most UTI data sets. And it seems to be associated with a very unique set of metabolites, a numerous and unique set of metabolites. The negative association network is equally as informative, but also not super informative if you just look at it in this way, except that UPEG has its own a unique negative association network as well. We can extract from this network, say, the beneficial lactobacilli, and we can look at what metabolites they seem to associate with. Interestingly, it's a lot of lipids. They seem to associate with a lot of urinary lipids like diacylglycerols, triacylglycerols, EPA's fatty acid, another triacylglycerol, Lactobacillus gasseri, johnsoni, crispatus, and inners. And it seems that gasseri has a relatively reproducible, unique metabolite association network in our data set that we don't fully understand yet. But even in the negative association network, it has its own unique set of compounds that it correlates with. Crispatus and johnsoni seem to be relatively interconnected, as well as inners. And this data has moved further, and in the lab we're looking at lactobacillus oxytrophies in this space, and a lot of these metabolites seem to fit in the theme of lactobacillus and what we're discovering about lactobacillus, data that Dr. Denisko has presented to this group before. So what we're finding is that it seems that the biochemical ecology of the urobiome is detectably associated with the microbial ecology. And we're starting to use this to try to understand what are nutrient niches, are there gradients in the bladder or in the microbiome. We also find that UPEG seems to be very strongly associated with its own metabolic network, its own metabolic signature. And within that metabolic signature, we're starting to see some important, perhaps, biomarkers that might be useful. So the last question that we asked with this data is a more translatable one. And there's two things that I want to discuss here. One is the diagnostic biomarker suite, and the second is a prognostic biomarker suite that we are finding evidence for. So if we just ordinate all of these patients based on their biochemistry of their urine, we find that the infected group is pretty strongly kind of uniquely clustered. And it clusters along features that are mostly lipids again. We're seeing a lot of lipids that seem to be discriminating in this data set. During infection, the urine seems to accumulate lipids. So we can do just a simple differential enrichment analysis of metabolites in those women that have an active UTI and those that have never had a UTI in their life. And we see that a lot of lipids are the distinguishing features that appear to pop up enriched in those women with active UTI. There we go. So we created a hypothesis that just basically stated that elevated urinary lipids are a metabolic signature of active infection, and how can we test it with this data set? So we looked at the summed lipids across the whole, just summed the concentration of all the lipids across the data set. And we didn't really, I mean, this is a statistically significant difference, but it's not terribly convincing in terms of its effect size and perhaps its biological significance. So we only found a mild difference. So we refined our question a little bit, and instead of summing all the lipids, we did a little bit of a fancy statistical approach to pull features out that are discriminating. This is called elastic net regularization. And we found an 11 lipid model that was very, very good at distinguishing infected from non-infected samples in the data set. We did a bit of cross-validation of this model within the same data set that it was discovered in. So this is just showing how well it performs within the data set. We found it has, within the same data set by cross-validation, an AUC of approximately 0.9 for identifying women that had an active infection. And a bit of biological interpretation of these findings. The lipids seem to correlate very strongly with known neuropathogens. So here's the most important lipid in the data, which is diacylglycerol. And it correlates quite strongly with Escherichia coli abundance. And the last little bit of data that I want to share with you is that we had 19 patients in this data set who were actively infected that had very reliable data for the time it took for them to present with a second infection, the time to recurrence over a 1-year follow-up. And this is a very good setup for us to do a hazard model. In this case, I did a Cox hazard model. And what I found was was 12 metabolites that were very strongly associated with essentially time to recurrence. And the top one here I want to focus on is deoxycholic acid. And deoxycholic acid is actually a microbial metabolite of a host primary bile acid. And what we found was it was quite strongly associated with time to recurrence. If we do just a simple Kaplan-Meier analysis, dichotomizing patients by the about the median into high and low groups, what we find is that these patients that have high deoxycholic acid in their urine actually recur quite a lot faster than those that have low. And here that is graphed out here in just an endpoint summary, showing that the low patients of low deoxycholic acid have above a 200-day median recurrence-free days, and those that have high levels of this deoxycholic acid have below 100. So we're developing this more now into seeing if this is validatable in a second cohort. I really want to stress that we have to validate this in a second cohort for these to translate these towards something that is actually meaningful. Hopefully they do validate. But another important question for us is, what's the source of deoxycholic acid? It's a microbial metabolite. Is it made locally, or is there something going on with some type of gut involvement and some being filtered from circulation that we don't really fully understand how it relates to recurrent UTI yet? So we found a subset of lipids that were diagnostic. This is important because diagnosis of these types of infections is not the easiest thing. Current dipsticks have about a 50% specificity. So we need novel molecular markers in this space. And we need markers to help us move forward towards a more personalized approach to therapy. This lipid signature seems to correlate very strongly with Escherichia coli, but other pathogens in the cohort might have unique signatures, and we're just not sampled in that space. And we also see that deoxycholic acid is a strong candidate to have prognostic appeal for recurrence. So our future work is moving from looking at the urine to looking directly at the tissue. And I just want to do a little plug for what we're doing now, which is using mass spectrometry imaging to directly image the bladder and to understand the spatial biochemistry and how that relates to the local microbiome. More on that is hopefully to come soon. So my lab is awesome. Dr. Nicole Gilbert has been part of this community for a long time. And, wouldn't be able to do anything without her and our funding. And with that, I'm happy to take any questions.